Okay, so this is where we left in the last class. We introduced you to the vast repertoire of membrane lipids, and you know most of the lipids are synthesized either in ER or Golgi and then transported to the plasma membrane with some lipid transport proteins. And I also mentioned that the different parts of a cell, for example, plasma membrane or ER or Golgi or even late and early endosome, has very distinct lipid composition, and I mean, this distinct lipid composition has uh, very serious implications in the physiological processes that happens in this particular organelles. For example, I told you that the mitochondria has a special lipid called cardiolipin, which is involved in some of the mitochondrial function that is not found in either plasma membrane or any other organ. Okay? Even nucleus has some of very interesting lipids. All right. Then we briefly touched upon what are the typical phases that are encountered in lipid bilayer membrane, and most commonly the phases that can be identified are a fluid phase or L alpha phase, and another phase is called the gel phase or L beta phase. So L alpha and L beta are the terminologies borrowed from liquid crystalline or liquid crystal field. So as I mentioned also that some of the features of the bilayers are very similar to the liquid crystal field. Uh, can you all hear me now? I saw a message that it's not audible properly.
At least I can hear, so I don't know about the participants.
Okay,
So
so as
yeah,
you can hear means others should also be able to hear.
I hope so. I hope
Yeah.
so.
All right.
It's
So
only thing if they are connected with the campus, so campus network connection that may have a intermittent disturbance. Yeah,
okay.
yeah.
So, although I mentioned that there are two phases that are very routinely identified in pure lipid membranes, for example, I take DPPC or di palmitoyl phosphatidylcholine or POPC, palmitoyl oleoyl phosphatidylcholine, I'll be very easily identify their phases. But as Dr. Santosh was asking in the last class, that in a living cell membrane is not made of only one kind of lipids, right? So there will be all different kinds of lipid with their individual phase properties, and that is also going to be influenced by protein. So the question is, will we be able to identify a phase transition in a, let's say, living cell? The answer is yes or no, depending on what kind of cells you're talking about. In some sense, you might be able to get a phase transition temperature, but in most cases, what will happen is coexisting phases, and it will not be either a pure L alpha fluid phase or a pure L beta gel phase. Most of the time, depending on the concentration of cholesterol in the membrane, we'll have what is called liquid ordered phase. So this is something between, you know, uh, the complete fluid phase or a complete gel phase. Hence, the properties of the lipids or the proteins will also be somewhere in between the fluid and the gel phase. Okay, this is something that we have to keep in mind. Although the bulk lipid composition will be uh, will be changing depending on the growth temperature if the organism is a poikilothermic. That means they cannot maintain their body temperature. It has been very well established for certain organisms, and in fact, it has been very well studied for yeast. And if yeasts are grown at different temperature, their lipid composition will be altered such that the membrane, a certain level of membrane fluidity is maintained. Uh, we also briefly discussed that the phase transition can be very easily determined by a thermodynamic process called the differential scanning calorimetry or DSC in short. But there are other indirect spectroscopic methods which will also allow us to determine the phase transition temperature. Now, or characterize the phase of the membrane. Uh, One of which I will discuss briefly, but just to mention few techniques. which are applicable for determining the phase transition other than DSC is, of course, fluorescence spectroscopy through polarization or anisotropy. In a similar fashion, we can also determine the phase, or sometimes it's called order parameter, by either ESR, that is electron spin resonance, or NMR, or that is nuclear magnetic resonance. Now, although we will be able to determine the order or phase transition temperature by, let's say, fluorescence, NMR, and ESR, as I mentioned earlier, that they are actually on different time scales. So that means the phase or the dynamic feature associated with different phases of membrane is not actually a transient phenomena, but actually maintained over a wide range of time scale. And as I already mentioned, that ichthyoramic organisms that cannot control their body temperature must adjust their lipid composition during cold stress to maintain a certain level of membrane fluidity in order to function properly. And this phenomena has been referred to as homeoviscous adaptation. Not, it's not necessarily that it has been understood properly, but this phenomena has been observed, and certain work has been done, and people are still trying to find out what are the protein sensors in the membranes which will be sensing the change in temperature, and that sort of modifies the metabolic processes so that one the proper lipid with appropriate saturation will be synthesized. Now, I just want to give you a glimpse of lipid dynamics through two very simple movies. And most of the time, although we know that the lipid molecules are dynamic, but we cannot appreciate because to us they appear as static, right? And if we take care of all the different uh, modes of motion, and they can be very nicely integrated into molecular dynamic simulation. So I'll play two movies just to give you an idea that how lipids are actually uh, exhibit dynamics in the bilayer plane, and these dynamic features are actually crucial for functioning of membrane proteins. So what we can see here in this ugly simulation, which is basically a sort of uh, conceptualization of all the different kinds of motion in nanosecond time scale, because the contemporary molecular dynamic simulations can only be carried out up to few nanosecond. Now they can be extended up to millisecond. But the idea is that they can give you a sense of the dynamics of the lipid molecules. Okay, so similarly, if we in this movie also, where we can see how the lipids are actually uh, exhibit lipids are exhibiting some dynamic feature, basically rotational and translational motion. If you look carefully, you will see that the lipids are actually changing their position. Okay, I'll say. And so two kinds of motions can be very easily identified from this simulation, that is the rotation and translation. Sometimes you'll also see the tails are wiggling, that's because of the uh, vibrational motion. So collectively, the translational motion, the vibrational motion, and the rotational motion defines the fluidity of the membrane. And fluidity, as I told you, is a term which is often used and sometimes even misused. Is very simply is inverse of viscosity. That means when the viscosity of membrane is very high, naturally fluidity will be low, and vice versa. When viscosity is low, fluidity will be high. So technically, it is taken as inverse of viscosity, but in fact, in earlier literature, you would find a term called microviscosity of membrane. That is, analogous to what is known as bulk viscosity. So basically any probe, any spectroscopic probe, be it a fluorescent probe or a ESR probe or NMR probe, will have certain kind of order parameter. And similarly, these probes can also be taken into bulk solution, like let's say glycerol, whose viscosity is known, and then based on the known viscosity values, membrane viscosity can be extrapolated by some spectroscopic measurements. Now, it used to be called microviscosity. If you go back to very old literature, you'll find that. But then later, people realized that the the bulk viscosity may not be exactly replicated in the sort of microscopic viscosity. So gradually in the literature, the microviscosity term in the membrane field sort of, I will say disappeared, but people stopped using that term. So, older literature you may still find it, new literature you won't see that. So keep that in mind. Uh, Now, as I mentioned that predominant dynamic features of the lipid molecules in the plane of the membrane can be categorized into three ways, mostly. Four ways. I'll come back to the fourth one. So one is vibrational motion. The second is rotational motion. The third is translational motion, or mostly what we call it as diffusion. Now, these are nothing new and nothing specific for lipid molecules. Any molecule will have 3 degrees of freedom that we know that. One will be vibrational degrees of freedom, one will be rotational degrees of freedom, and another one will be translational degrees of freedom. So I mean, why wouldn't they work for lipids? Then that's why the lipid molecules are constantly exhibiting these dynamic features, and they also influence the dynamics of the proteins that are embedded in the membrane bilayer. Vice versa, protein will also influence the dynamic features of. Sorry, is there a question?
No,
Okay, all right.
it was some sort of humming from the back.
In fact, when I come to what is called lipid-protein interaction, we'll talk a little bit about how proteins influence the dynamics of lipid and lipids influence the dynamics of protein. We already know that because the moment I am talking about fluidity, that is basically how collective membrane dynamics, that means the lipid dynamics, will modulate the dynamics of proteins. In fact, this has been thought of as one of the predominant factors by which lipid bilayer would modulate the function of membrane proteins. But gradually people realize that it's not only fluidity; there are other properties, and as I defined in the last class, they are called emergent properties. I'll come to that in a moment. So apart from vibrational, rotational, and translational or diffusional motion, there is another motion that lipids exhibit, and that is called a transverse diffusion. So that means one lipid molecule can come from upper leaflet to lower leaflet or lower leaflet to upper leaflet, and this is important for maintaining membrane asymmetry. And this motion is also known as flip-flop. You all may know that the membranes are asymmetric in the sense that the two leaflet will have different lipid composition. A very classic example of that is phosphatidylserine. It's a negatively charged lipid often found in the inner leaflet of the membrane. Usually that does not come out in the outer leaflet unless there are some pathological condition. For example, if the cells are going through apoptosis, and this is one of the classical marker for apoptosis to find out the exposure of PS in the outer leaflet. And it is also found in some cases, for example, in erythrocyte, if there is any, any disorganization in the spectrin, or you treat some spectrin destabilizing agent, PS also comes out, and it can be very easily identified by PS binding protein, that is annexin. And by annexin labeling, you can find out whether the PS has been exposed or not. So this asymmetry is actually maintained by very slow diffusion of lipids across the leaflet, and that is known as transverse diffusion. So usually the vibrational motions are in the picosecond to femtosecond time scales. Rotational motions are in sub-nanosecond time scale, whereas translational and diffusional motions mostly in microsecond to millisecond time scale. But transverse diffusion across the leaflet is actually even slower process. So sometimes it takes even minutes, hours, and days for for one lipid to come from one leaflet to another leaflet. The reason is when one lipid is coming to another leaflet, the head group is actually has to come through a hydrophobic environment, which is not thermodynamically a very stable situation. That's why it has a very active, very high activation energy barrier. There are specific enzymes. These are called flipases and flopases, and so they help in maintaining the transmembrane asymmetry by assisting in the flip-flop motion. So this is another slide which actually comprehensively depicts all the different kinds of lipid motions in a bilayer. This is actually taken from a very classic textbook in membrane biology by Genis, by Eagle. And in fact, if you are really interested in learning more about the fundamentals of bilayers and membrane physics, you should refer to this book by Philip Eagle, and there is a similar book by Dennis. I'll share the reference at the end of my class. So, all right, Santoshji, you are still fine with my voice?
Yeah, it's okay. It's okay to me.
Okay.
I think so. Yeah.
Okay. Yeah, great. So, the few motions that are routinely studied are, as I mentioned, that rotational diffusion, uh, lateral diffusion, or translational diffusion. In the faster time scale, bond oscillations and trans-gauge isomerization, it is in a very faster time scale, and and there are two motions which are identified. One is called the protrusion, where the lipids are actually, change their vertical position because they are constantly uh, uh, experiencing upward drag and downward drag. That's why they are, oscillating even in the uh, vertical direction. And these protrusions are collectively changing the membrane topology in the form of undulation, which since protrusions are in a faster time scale, so when uh, lot of protrusion motions are sort of combined, then we have undulation in membrane, right? And these are very important dynamic features in the sense of either membrane interaction. So later on, when we'll see membrane deformation and membrane remodeling, we'll discuss these forces even more strongly, because these are the forces which will prevent two bilayers to merge. That's why we always need a fusogen for merging of two membranes, or for fusion of two membranes. Fusion proteins are actually useful in sort of damping this motion so that two membranes can eventually be fused whenever required. Okay, we'll come to that during membrane remodeling process once again. Uh, so what I have been trying to say here is that there are different kinds of uh, local motions exhibited by the lipid molecules or part of the lipid molecules. But then these collective motions can actually modulate, physiological functions, because the small motions can now be extrapolated into a different spatiotemporal region or different spatiotemporal scales. So if you look at this particular chart here, where whereas the smaller or sort of local dynamics, for example, side chain rotation, electron ion transfer, which happens in a very faster time scale, for example, in picosecond, also has a motional magnitude about only few angstroms. So that means they are very localized motion. But as you gradually increase the time scale of this motion, for example, local conformational change or domain motion, all the way from nanosecond to microsecond, you also see that the motional amplitude, that means the repercussion of those dynamic feature, is also propagated over a wide range of wide range of spatial scales. Similarly, even if we go into a higher temporal domain, for example, millisecond to microsecond, the motional magnitude has also increased all the way up to 100 angstrom. So, in a sense, the local motions are actually combined to bring about larger dynamic changes over a wide range of spatiotemporal scale, and that is important in many physiological processes in membrane, for example, signal transduction. So signal transduction is a process which is a, you know, uh, basically a cascade of events happens over wide range of spatiotemporal scale, starting from electron ion transfer, side chain rotation, to local conformational change, then domain motion. If it is involved in a ion channel, then a channel opening, and then a particular signal which has been accepted by a receptor molecule will be transduced across the membrane into the cellular interior. Okay? So for that to happen, every component, every dynamic component of the membrane has to work in concert. Here is again a comprehensive chart in which I'm trying to show that the the overlapping of the time scales of different motions that have been identified in uh, bilayer and the corresponding spectroscopic or microscopic technique for interrogating those motions, right? Now, although we are talking about these motions in the context of lipid bilayer, but they can be applied to proteins as well, right? Even the proteins are dynamic too. Even the proteins are dynamic too, right? So, for example, if you think of if if you think of vibrational motion, and that can be studied by Raman or IR spectroscopy. Similarly, if we are interested in trans-Gauss isomerization, that will be reflected in fluorescence polarization and fluorescence solvent relaxation. Lipid rotation again can be studied very well by fluorescence depolarization or fluorescence anisotropy. Some of the larger motions, for example, larger domain motions or lipid flip-flop, can be studied very easily by NMR, NMR hydrogen deuterium exchange, or even a uh, uh, analogous version of fluorescence anisotropy, that is phosphorescence depolarization or phosphorescence anisotropy. Um, NMR relaxation can be used to study the motions of over a wide range of time scales, all the way from picosecond to you know an hour time scale, and we all these things can be complemented by molecular dynamic simulations, either all atom molecular dynamic simulation or to a slightly larger uh, uh, time scale in using a coarse grain molecular dynamic simulation. So depending on the motion of the or the dynamic feature of the lipid molecules that we are interested in, we can choose a particular spectroscopic or microscopic method to interrogate that particular feature of the membrane. Now, I am sure that you all are familiar with fluorescence or fluorescence anisotropy. But just to give you a brief description about what we can do with fluorescence microspectroscopy as far as membrane dynamics is concerned, is that fluorescence spectroscopy per se, uh, and particularly fluorescence anisotropy and polarization, is a very good tool to study the rotational dynamics of the lipid molecules, or even the rotational dynamics of the protein molecules as such, or part of the protein when they are embedded in membrane. As you might know that the light, as it is coming out from the light source, is depolarized. That means it is actually has electromagnetic oscillation in all possible directions. But when we make this light to go through a polarizer, then we'll have light only in one particular direction, and that is called plane polarized light. Now, when this plane polarized light is subjected to a fluorescent molecule, the molecule will absorb the light only when the molecular transition dipole will be aligned with this polarized excited light. Now, let's say this molecule is actually oriented with the same direction of the polarized light. It will be excited and will maintain that polarization. What does that mean? That if we keep the analyzer, which is nothing but another polarizer, in the same direction as the polarized excitation light, we will get intensity. On the other hand, if the protein or the lipid molecule is exhibiting a very fast rotational motion or tumbling motion, what will happen is that the direction at which the molecule absorbed will no longer be maintained because the molecule is constantly changing its orientation. So in that case, what will happen? If the polarizer is kept in the same orientation with the excited light, no intensity will be detected. So basically, if we take the ratio of the intensity of the light at two different orientation of the analyzer. In one case, the analyzer, which is nothing but another polarizer, is oriented with this polarized light, will get intensity. If we take it to a completely orthogonal direction, and we may not get any intensity because the molecule didn't rotate at all. On the other hand, if the molecule is tumbling in a very fast time scale, then what will happen? All the directionality will be lost and all the intensity will be found in the perpendicular or orthogonal direction. So if we simply take the ratio of the two intensities, that two different polarizer analyzer combination, we will be able to capture the rotational motion of the molecules, be it a protein or be it a lipid, in bilayer environment. And that ratio is sometimes referred to as fluorescence anisotropy. In some literature, it is referred to as fluorescence polarization. They're the same thing, except there is a small arithmetic difference. So, since the molecules are constantly rotating, we can actually find out the rotational diffusion time from an experiment called anisotropy decay, which is nothing but anisotropy over the fluorescence lifetime, and which is nothing but nanosecond times time. Remember that fluorescence depolarization, as I mentioned, is in the nanosecond time scale, and in the nanosecond time scale, we have the rotational motion, the transverse isomerization. So by using fluorescence anisotropy, we can very easily find out what's happening in the rotational motion of the lipids in a given membrane situation. This Actually, from this particular experiment, we can find out not only the total anisotropy, but also the rotational correlation time. So the anisotropy will give us the idea about the microviscosity of the or the fluidity of the bilayer, whereas the rotational correlation time will give us the idea about the rotational mobility of the lipid molecules or part of the protein molecule sometimes. All right. Now, as I mentioned that uh, fluorescence anisotropy, and there is another technique which is also routinely used, and I will not have enough time to take you through that, and that is called solvent relaxation. Solvent relaxation is actually relatively faster technique than fluorescence anisotropy, but we do not get the same kind of information that we get by fluorescence anisotropy. So, actually using solvent relaxation, we can find out more about the head group mobility of the lipid molecule, because these are the region which responds to the uh, change in excited state dipole moment. So based on these features, we can actually find out the uh, the dynamics associated with the lipid head group, whereas by fluorescence anisotropy or polarization, we can more or less find out about the dynamic feature about the acyl chain region of the membrane. Okay, so solvent relaxation provides dynamic features of the lipid head group, whereas the anisotropy, fluorescence anisotropy provides dynamic information regarding the hydrocarbon chain of the lipid bilayer. But these are all local motions. But uh, what about some global motions that are found in the membrane? Meaning, what about the translational motions or diffusional motions in membrane? And how are these diffusional motions are studied in membrane? There are primarily two techniques. There is a third technique that is also used, and in fact, I mentioned it in the last class. That is SPT or single particle tracking. I'm not going to go through the detail of that, but I'll briefly discuss about what is FRAP and what is FCS. FRAP is nothing but fluorescence recovery after photo bleaching. So I'll take you through a small movie which will explain what exactly done in a FRAP experiment. This movie has been produced by the lab of Jennifer Lippincott-Schwartz. Some of you may know that she is one of the pioneer in FRAP and quantitative cell biology.
The lateral mobility of membrane proteins
So let me know
measured
if you can hear, Santoshji, can you hear the voice in the movie?
Yeah, I can.
Okay.
the
I think
lateral
there's been complaint with some of the students that the voice is too low, but I'm surprised. Like at least
So
I'm able to hear clearly. So
okay,
uh,
I got
either
it.
we have to bear or like see if anything can be done at your end. That's the request. Yeah.
All right. Okay, so I'll play a small movie here just to explain what exactly done in FRAP.
proteins can be measured in living cells by FRAP, which stands for fluorescence recovery after photobleaching. For this purpose, membrane proteins are often expressed as fusion proteins with the green fluorescent protein GFP and observed with a fluorescence microscope. A selected area of the cell is then bleached with a strong computer-controlled beam of laser light. Those membrane proteins that are not anchored and therefore can diffuse in the plane of the membrane quickly exchange places with their neighbors and fill back in the bleached area. From the rate of this fluorescence recovery, the diffusion constant of the protein can be calculated. Here, GFP is fused to a membrane protein that lies in the membrane network of the endoplasmic reticulum. After bleaching, we observe quick recovery of the fluorescence, showing that the protein is very mobile in the plane of the membrane. The same experiment can be repeated using a protein that is firmly anchored and not free to diffuse. Here, we observe GFP fused to a protein of the internuclear membrane that binds tightly to the meshwork of the nuclear lamina. After photobleaching, no fluorescence recovery can be seen over the same time frame.
All right. So I think you got the essence of the FRAP experiment, and it's based on the principle of microscopic reversibility. That means when you sort of photobleach a lipid or protein molecule in the plane of membrane with a strong laser, you are not destroying their ability to freely diffuse. So you create a momentary darkness and then wait for the recovery. And the recovery happens in the sense that you'll get back fluorescence from that same bleach spot because the other molecules are constantly diffusing. So the molecules that have been bleached will come out of that bleached zone, whereas the new molecules which has not been bleached will come back to that zone, right? And so that that's basically the molecules are constantly diffusing through this particular uh, spot in the or this particular region of interest on the membrane surface. So from these experiment and the rate of recovery, uh, usually the tab data looks like this. You have a small, very sharp change in the intensity, and then a slow recovery. Sorry about that. A slow recovery of intensity. And based on these curve, we can actually find out two parameters. One is diffusion coefficient, which is nothing but a quantitative parameter which describes the diffusion of lipid or protein, and also another parameter that is called mobile fraction. That means what is the fraction of a particular molecule of interest is actually mobile in that time scale, and which has very significant repercussion. For example, some of the lipids and protein molecules may not be available compared to other molecules. There could be a number of reasons. A, they may be some of the molecules, or be it a lipid or protein, may be in a different microscopic domain, or they may be sometimes anchored with the spectrin or actin, depending on which cells you're talking about, and that's why they're less mobile. So this information will be available by a FRAP experiment. An analogous experiment has been devised and again very widely used in not only in the membrane biophysics or cell biology field, but also in the in protein aggregation field, and that is known as FCS or fluorescence coordination spectroscopy. So, what happens in fluorescence coordination spectroscopy is, by the way, it is also known as one of the single molecule spectroscopies, that we create a very narrow zone in the membrane by exploiting the confocal microscope. So, and we observe fluorescence intensity from this very tiny zone, which is nothing but the confocal volume, and in the order of 1 femtoliter. Now, the advantage of watching fluorescence intensity from a very tiny volume is that we will be able to detect the coming in and going out of a single molecule. So if we are looking at a bulk environment where there are, at least, even in the sub-nanomolar concentration, there will be too many molecules to count. But if we restrict our observation volume into very tiny volume of, let's say, 1 femtoliter, we'll be looking at only 1 or 2 molecules at a time. Now, what is the advantage of that? The advantage is that we can actually identify when another molecule is coming in this confocal volume, or another molecule is going out, because we will be constantly looking at the fluctuation in the fluorescence intensity that is coming out from this tiny volume. So based on this fluctuation, we can again calculate something called an autocorrelation function given by G as a function of T. And from this autocorrelation function, we can find out diffusion coefficient, because the molecules are moving, that's why they must be associated with a diffusion coefficient. But unlike mobile fraction, in case of FCS, we'll be able to find out the local concentration of the molecule, because we'll be able to count the number of molecules that is present in, let's say, 1 femtoliter volume, and then we can, depending on the size of the cell, we can extrapolate that how many different molecules are present. So we can actually find out the local concentration using FCS. Sometimes FRAP and FCS provides complementary data. So one thing we have to keep in mind that although FRAP and FCS provides data on the similar information, on the similar phenomena, the value of diffusion coefficient of the same molecule determined by FRAP and determined by FCS may not be the same. The reason is FRAP and FCS may be looking at the different special region on the membrane, right? Although both are confocal based technique, but FCS is looking at the diffusion in a very tiny region, whereas FRAP looks at diffusion over a slightly bigger region of interest. And that's why in case of FRAP, more than one kind of dynamic features are captured, and that's why the diffusion coefficients will be slightly larger, whereas in FCS, it's very local diffusions will be captured. So, that's why the diffusion coefficient determined by the FRAP and FCS may differ because of these reasons. All right, so after having some idea about some of the dynamic features of the plasma membrane. Let's now get back to the plasma membrane as such and where we started to. So, as I told you that the basic idea of plasma membrane is based on fluid mosaic model. But that model basically says that there are plenty of lipids in it, and it constitutes a two-dimensional fluid. Okay, I'll come back to your question, Santoshji, later.
Yeah, sure. Yeah, thank you.
Yeah.
Uh-huh.
So, according to the fluid mosaic model, which is nothing but the two-dimensional anisotropic fluid made of phospholipid molecules in which proteins are embedded either in transmembrane fashion or in peripheral fashion. Fine. But then people realize that that is not the exact case. So what fluid mosaic model tries to project that there are, huge amount of lipids and proteins are freely diffusing. In other words, the membrane is mostly made of lipids and proteins are put in the form of mosaic. However, that is not the case. So in fact, it has been found for some of the cells that lipid to protein ratio is actually 1:1. So that means maybe per one protein molecule there may be one lipid molecule. In other words, membrane is much more crowded than it has been perceived in fluid mosaic model. So in such a crowded situation, one protein will definitely influence the function of another protein, and most importantly, lipids will influence the protein function of membrane protein. So, uh, as although fluid mosaic model was able to explain many of the features, some of the features that are arising because of this molecular crowding could not be explained in fluid mosaic model. So that's why the model is ever evolving, and the concepts of lipid phases and their significance, whether transient or non-transient, and non-bilayer structures are all now being taken into account in explaining the membrane phenomena, which were not present in fluid mosaic model. And most important of that is the rat hypothesis. So now the proteins which were freely diffusing in fluid mosaic model or according to fluid mosaic model is now restricted in a very small region of the membrane because these are now these membrane are now domainized. That means the composition of this particular membrane at this point may not be same, at another point in the same membrane. And because of the restricted diffusion. In fact, Santoshji has brought out a very important point, and I'll get back to that towards the end of my lecture today. One of the things that we have to appreciate is that although we said that the plasma membrane is mostly made of phospholipids, and most of these phospholipids can be thought of as if a molecular cylinder, and they can very nicely pack into a bilayer fashion. And since the bilayers have exposed edges, the moment they come in aqueous environment, they will try to wrap around themselves to form a closed structure like vesicle. Unlike micelles, which will form more of a spherical monolayer kind of structure. Now, depending on the structure of the lipid molecules, some of the lipids may not be very happy in a purely lamellar structure, and they will tend to deviate from the typical lamellar structure, provided they have the ability to phase out from the existing membrane. But most of the time, this is not possible because, as I told you earlier, that when we are dealing with a laboratory system where we have a pure lipid, we can get into different phases very easily. But when we are dealing with biological membrane, which is made of different kinds of lipid, each lipid has a different preference. And when these preferences are not satisfied by the constraint and brought about by some proteins, what will happen is a stress will be introduced in the membrane, and that is known as curvature stress or curvature frustration. And this manifests, and this is basically nothing but a stored potential energy, and this potential energy will be used in membrane remodeling processes. In fact, some of the membrane remodeling processes that we'll see later on during fission and fusion is facilitated by the lipids which actually do not support the lamellar structure. So they make this lamellar to non-lamellar transition easier because of this curvature stress. Now let me make this point more clear in this slide. So let's take the example of POPC lipid, which is a very representative mammalian lipid, that is palmitoyl oleol phosphatidylcholine. That means it has two SI chain, one with one unsaturation. And this particular lipid molecule can be thought of as a cylindrical molecule, and they can very nicely pack into the lamellar structure or bilayer structure. But let's look at some of the other lipid molecules. For example, POPE, which is palmitoyl oleoyl phosphatidylethanolamine. Now, between phosphatidylcholine and phosphatidylethanolamine, phosphatidylethanolamine has a slightly less area in the head group, such that this molecule can no longer be thought of as a cylinder, but can be thought of as inverse cone. Now, when these kind of molecules pack, their natural tendency will be to pack in a particular orientation, and that is known as inverse hexagonal phase or a non-Bilayer phase. Unlike phosphatidylcholine, which will always form a lamellar bilayer phase, phosphatidylethanolamine under certain condition will form an inverse hexagonal phase. Similarly, cholesterol by itself, if taken under appropriate condition, will also form inverse hexagonal phase. Likewise, if we take lipids, which is more like detergent, for example, lysopc, one of the favorite lipids of Santoshji, because he has been using it very judiciously in arresting cells at different fusion stages. So the lysopc, on the other hand, inverse hexagonal phase, it will form what is called a hexagonal phase, or if you think of is a micellar phase. If you take a cross section, this is nothing but micelle, because this now, because of the one tail in the lysopc or lysophosphatidylcholine, it can now be thought of as an inverse cone molecule, and which can now be very easily packed into a hexagonal fashion. So the point is, depending on the shape of the lipid molecule, they have different preference for different kinds of non-bilayer structure. And this ability of the lipid molecules to deviate from the lamellar structure is quantified by a parameter called spontaneous curvature, which can be very easily determined by a small angle X-ray scattering. For example, POPC has a very negligible negative spontaneous curvature, negative 0.022. So that's why it's formed or prefers to form a lamellar structure. Whereas POPE and cholesterol has very high negative curvature. On the other hand, lysopc has a positive spontaneous curvature. So these features actually modulate the shape of the membrane, because or contribute to the remodeling energetics. Or when, for example, two membranes are trying to fuse, they have to deviate from the lamellar structure, either going through a stalk phase, which has similarity with the inverse hexagonal phase, or the hexagonal phase will be stabilized if the lipid compositions have POPE, cholesterol, or lysopc. So when we'll be talking more about fusion and membrane remodeling, I'll bring out this point again, that the ability of these lipid molecules to deviate from the lamellar structure will facilitate membrane remodeling, of course, with the help of proteins. Now, let's switch gear a little bit and talk about membrane proteins and their orientation and some of their properties. So as I already mentioned that, already approximately 30% of a typical proteome would be membrane proteins, and of which 60% will be drug targets. And these numbers are increasing day by day. But one of the features of the membrane proteins that, unlike soluble protein, which can be very easily purified and crystallized, membrane proteins are very difficult to purify, because it will be difficult to remove all the lipid molecules and then reconstitute it in a suitable environment. The secondly is, for the same reason, it will be difficult to crystallize in pure form or in native condition. Nowadays, you might be aware that a lot of membrane protein being crystallized, and for example, in several years back, one of the very important class of membrane proteins, that is G protein receptors, have been crystallized by Brian Kobilka and Robert Lepkowitz, and they were awarded Nobel Prize not only because the GPCRs are important class of molecules, GPCRs are also membrane proteins, and they overcome this difficulty of crystallization of membrane proteins by employing a technique which again exploits the lipid biophysics. So the only limitation is that, and I think we have to accept that limitation, is that the membrane proteins are not necessarily crystallized in their native conditions, unlike soluble proteins. So there is something called a lipidic cubic phase, which supports the membrane protein crystallization, but lipidic cubic phase is not a phase that is found in physiological conditions. So we have to accept that limitations for the time being, till we, till the techniques of membrane protein crystallization advances to a level where the proteins can be crystallized in their native condition. One of the features of these membrane proteins crystallization is that all the dynamic informations are lost, because as I mentioned earlier, that in order to understand the function of a membrane protein, and in fact it is also relevant for soluble protein also, we have to understand how the dynamic feature of this particular protein, right? So although we need crystal structure in order to understand the molecular details, but that crystal structure has to be complemented by the dynamic information regarding the functioning of the protein. And these dynamic informations will come from spectroscopic and microscopic measurements. So first challenge in studying membrane protein is to purify them and to crystallize them. And the second challenge would be to understand their dynamic feature at native condition using various spectroscopic and microscopic techniques. If you are interested in the progress of membrane protein crystallization, membrane protein database, this is a site which is actually the website of Stephen White in UC Irvine, and this is one of the most updated websites which talks about the development of membrane protein structure, membrane protein crystallization, and some of the physical properties of membranes that are very useful. For example, a very good bioinformatic tool for identifying bona fide membrane protein is Hydrophobic Plot. Some of you may know that. So, for example, if we have a protein sequence and we find a long stretch of hydrophobic residues at repeated interval, this is telling us that this protein or this putative protein is likely to be a membrane protein. So this idea of estimating the hydrophobicity of amino acid is most reliably can be found out by some of the techniques developed by Stephen White and are made freely available in this particular website. So again, I urge you to look at this particular website whenever you have a chance to. Now, as I mentioned earlier in the introduction of fluid mosaic model, that Singer-Nicholson model also distinguishes between two different kinds of membrane protein. One is called a peripheral membrane protein, which is basically associated with only one leaflet of the membrane. And most of the time they can be very easily separated from the membrane by simply changing the salt concentration or other buffering conversion. The other kind of proteins which are more important is the integral membrane protein. Integral membrane proteins are embedded in the membrane and they are in intimate contact with the lipid molecules. So for soluble proteins are always in contact with water, whereas membrane proteins are solubilized by lipids. So the water molecules of soluble proteins are replaced by lipid molecules. So these, you can imagine that these particular transmembrane helix sequences have to be extremely hydrophobic in order to be stable in the lipid bilayer. So transmembrane proteins are, you know, not only alpha helical, but there are a class of beta barrel proteins. For example, bacterial codings are all beta barrel proteins. In fact, another classic protein which is a beta barrel protein is VDAP, and voltage dependent anion channel, that is found in mitochondria. So that is also a beta barrel protein. Now, usually membrane proteins are either alpha helical or beta barrel. They don't have much of, unlike soluble protein, part of it is beta or part of it is alpha helical. That rarely happens in case of membrane proteins. Now, one of the features that we have to appreciate here, since the transmembrane domains of the integral proteins are completely embedded in the lipid bilayer environment, so naturally the physical properties of the lipid bilayer will be a crucial factor in the functioning of this membrane protein. Now, one of the crucial factors that we have been discussing is fluidity. Other than that, there are properties which emerge because of the bilayer fashion or bilayer arrangement of lipid molecules. And these are, I'll come back to this slide later. Just since I was telling you already, these are membrane fluidity, membrane curvature or elastic property of the membrane, and the extra potential energy that is stored in the form of curvature stress will also modulate the membrane protein function. The thickness of the bilayer, which will manifest in the form of hydrophobic mismatch, I'll come to that point later, and membrane electrostatic properties. So membrane electrostatic properties will contribute to the membrane potential in the form of dipole potential, transmembrane potential, and surface potential. And all these three forms of electrostatic property of the membrane are very crucial in supporting the activity of membrane protein. In fact, I will take you through one particular example of membrane potential from my own research, just to highlight that how physical properties of membrane actually modulate the function of a membrane protein. Other than that, there will of course be specific effect, that means which involves chemical binding of the lipid molecules into the protein structure as a binding motif or sometimes as a cofactor. So these are more specific effect where one particular protein, one particular kind of lipid is required for the functioning of one particular lipid, one particular protein. For example, cholesterol is required for serotonin 1 receptor, and it has been very recently found that there exists a binding motif called track domain where cholesterol molecule can go and specifically bind. And so this binding is necessary for the functioning of the serotonin 1 receptor. Sometimes for many other proteins, these changes are also allosteric. So that means lipid binding brings about a global change in the protein structure, and that is required for the function. So these comes under the specific effect, and other than that, the general properties of the lipid bilayer membrane in the form of membrane fluidity, curvature stress, membrane thickness, and membrane electrostatic property are influencing, and these factors are common for all kinds of proteins. So, if for any situation, if the membrane composition is changed, the functioning of membrane protein will be influenced by any of these properties, right? Now, what is most important is that the specific effect and general effect of lipids are not mutually exclusive. That means a membrane protein which has got a specific binding motif for cholesterol, that does not mean that this protein will not be influenced by the other attributes of cholesterol. For example, cholesterol has a profound effect on membrane thickness in the form of dominization, right? So the liquid ordered phase that I was mentioning in the last class will have a higher thickness compared to liquid disordered phase. And this will bring about a situation called hydrophobic mismatch, which will have a influence on the membrane protein conformation. So just because some protein has got a binding motif for a particular lipid, it will exhibit specific lipid effect, but it may also exhibit general lipid effect. Okay, so that these two effects are not mutually exclusive. Since I'm here in this slide, I might as well show you that membrane lipids in the context of protein can be categorized into three types. One is called bulk lipid, and all the specific effects, sorry, all the general effects that I discussed in the last slide are actually mediated by this bulk lipids. So bulk lipids are how do we identify bulk lipids? Because as the name suggests that they are available in abundance, that's their bulk, and most importantly, they are free to diffuse. So they have a very low residence time on the protein surface. So they may be accidentally found on the protein surface, and if you have the ability to monitor the lipid molecules, then you may find that these proteins are, spending some time on the protein surface, but then again in the next moment it will be exchanged by another protein, another lipid, because that lipid is also freely diffusing. How do you know that? Again, it can be very well studied by ESR and NMR, because when the lipid molecule is bound to the protein molecule, it will be slowly, its dynamics will be retarded or slowed down. That can be captured by ESR or NMR spectroscopy. On the other hand, when the molecule is free from the lipid protein surface, its dynamic features will be much faster, which will again be very conveniently detected by NMR and ESR. So just by looking at the NMR or ESR line signal of lipid molecules, we can identify whether they are bulk lipid or they are annular or non-annular lipid. I'll come to annular lipid or lipid shell. So what happens is most of the proteins or most of the transmembrane protein has a immediate vicinity, or the lipids in the immediate vicinity are known as annular lipid, and these lipids are held slightly tighter than the bulk lipid. So they exchange rather slowly than the bulk lipid. So again, by the same kind of experiment, we can find out whether they are bulk lipid or they are annular lipid by finding out their rate of exchange with the bulk lipid. So the bulk lipids will quickly exchange with each other, whereas the annular lipid will slowly exchange with each other, right? Because they are held more tightly on the surface of the protein. Now there is a third kind of membrane lipid, and that is known as non-annular lipid. Now the non-annular lipids are the most specific lipids which are found in the crevices and of a transmembrane protein. For example, let me just show you a real example. And this is a crystal structure of beta-2 adrenergic receptor, again determined by Brian Kobylka. And in that structure, they found that the cholesterol molecule is actually found between the interface of two dimers. For example, this is also shown here. So these are actually called the non-annular lipids. So these non-annular lipids are not, sorry about that, are not exchange. They are not going to exchange with bulk lipid or annular lipid. They are so tightly held that they are actually found in the crystal structure of the membrane proteins, right? And most of these lipids have specific binding motif on the protein surface. So the specific lipids are most likely the non-annular lipids. Okay, I actually skipped few of the slides in between, so I'll go back to that and in the context of membrane protein organization. All right, so we understand that there are peripheral membrane proteins, there are integral membrane proteins, and integral membrane proteins are more influenced by the physical properties of the lipid bilayer, either by a general effect or by a specific effect. Now, one of the interesting features of the organization of the membrane protein comes from the fact that different amino acids have different preference for aqueous environment versus hydrophobic environment. And this is again nothing new. That the whole idea about the protein folding is based on the fact that certain amino acids have preference for water and certain amino acids have have no preference for water, right? And the hydrophobic collapse is basically centered around this concept. Now, the same idea is applicable in the composition of the membrane protein. So, therefore, and this is actually from very old study, very early study of Charles Stanford. In fact, Stanford is the one who has worked tremendously in understanding the hydrophobic effect. He has written a book called The Hydrophobic Effect. And if you have the opportunity to get hold of the book, please read that. So, what Stanford and others have been trying to identify is that what is the energy associated with one particular amino acid if we wanted to transfer that amino acid from aqueous environment to our hydrophobic environment or vice versa. In other words, Stanford was interested in calculating the free energy transfer of single amino acid from aqueous to hydrophobic environment. This idea can be borrowed into membrane protein organization. So when some of the amino acids which are hydrophobic, they can very easily partition into membrane, whereas those which are extremely hydrophilic and charged may not be easily partitioning into membrane. So these factors will be membrane anchored glycoproteins can be classified as peripheral membrane protein. Yes, because the membrane anchor is most of the time is a lipid molecule and a fatty acyl chain. In fact, I have a slide and I'll show it to you. Thank you for bringing that. In this context, tryptophan plays a very dubious role because tryptophan is a hydrophobic molecule, but then tryptophan is also a polar molecule, right? It has got a permanent dipole moment. So the tryptophan then is a special case. I'll come to that later. And so based on these concepts, there are several hydrophobicity scale that has been defined or devised. Now, why are these hydrophobicity scales are important? Because see, if we want to look for a membrane protein or we want to find out whether a protein is a bona fide membrane protein or not, we have to find a window of hydrophobic amino acids. So that means we have to be able to define the hydrophobicity of amino acids. And then depending on what is your criteria of defining hydrophobicity, there will be different scales. And most of the time, these scales do not agree. For example, tryptophan in one of the scales will be found as most hydrophobic, whereas in some scales it will be found moderately hydrophobic also. So where do you space tryptophan, right? Similarly, there will be some other amino acids which will not be, which will not have a unique position in all the scales. So that's why there will be different scales that has been introduced, but the most reliably the scale that can be used most reliably or most widely used, as a matter of fact, is what is known as biological hydrophobicity scale or an octanol based scale. Again, that has been developed by Stephen White, and most of the hydrophobic plot, hydropathy plots are actually used by or uses that concept of hydrophobicity or hydropathy. Okay. So again, if you're interested, you can go back to the Blanco website and you'll find how that is exactly calculated. Um, so based on that, we can actually define a bona fide transmembrane domain. So mostly the protein, the amino acids which are bulky and hydrophobic, for example, isoleucine, leucine, valine, phenylalanine to a certain extent, will all be found in the middle of the membrane or middle of the transmembrane domain. Whereas the charged residues are mostly found in the uh, the interfacial region. The aromatic amino acids are actually plays a very important role. For example, tryptophan, tyrosine, and phenylalanine. Although they are hydrophobic, but as I said, since they are aromatic, they have a permanent dipole moment, so they are also considered as polar to a certain extent. So they are also able to interact with the interfacial region of the membrane. So, and most of the time, these aromatic amino acids are also used as an anchor for the transmembrane proteins to hold on to membranes. And this feature has been found in many membrane proteins. One prominent example of that is potassium channel. So the potassium channel or Shaker potassium channel has a belt of aromatic amino acid at the interfacial region, and these interfacial aromatic residues are very crucial for maintaining the orientation of the membrane protein. Right? Now there was a question about whether the lipid binding proteins are peripheral proteins or integral proteins. They are actually peripheral proteins because they are attached with a uh, lipidic domain. If they have a transmembrane domain, then they will be considered as a monotopic transmembrane domain. So since I'm in this slide, I might as well explain that integral membrane proteins can be defined as a single transmembrane uh, protein. They can be of multiple transmembrane protein like GPCRs, which usually have 7 transmembrane domains. And then, of course, the beta barrel proteins like porins or VDAC, the voltage dependent ion channel, which are usually as monomers of one unit. Sometimes they can also dimerize under certain conditions. How do we view transmembrane algorithm for transmembrane prediction? Yeah, so that's what I was saying, that you can go to Blanco and you will be able to see how the transmembrane predictions are done. I don't have the time to take you through that, but if I in the next class, if I have time, I will try to do that. It's basically you define a window of hydrophobicity and slide that window over the entire sequence of the protein and pick up the peaks, which gives you the maximum hydrophobicity. So those will be the likely transmembrane domain in a given protein. All right. Okay. So I explained that. So although I gave you the example of non-annular lipid, here I have two lists which actually uh, uh, depicts the examples that have been found in the literature where the non-annular, the annular lipid have been identified: aquaporin, datilorubicin, gramacidin. Gramacidin is a fairly small ion channel, and the methods that have been used to identify the um, uh, category of lipid, whether they are bulk lipid or annular lipid or non-annular lipid. Most of the time, they can be a spectroscopic method, for example, ESR or EPR or NMR. Sometimes they can also be found by crystallography, and here also this list also has the lipids that have been found for corresponding proteins and what are their attributes, what are their functional attributes as far as these proteins are concerned. So this is again a very comprehensive list. If you're interested, you can go through that. It's taken from this particular review. Similarly, the same review has another list which talks about the non-annular lipid. Remember I told you about the beta-2 adrenergic receptor, and it has cholesterol. So this is one of the non-annular lipids that have been identified by spectroscopy. Similarly, there are many other proteins where it can be found out. For example, serotonin 1 receptor is cholesterol, and it's an allosteric allosteric modulator, and it has been actually found out by pure biochemical methods. So no spectroscopic methods or no crystallographic method, although later it has been proved when the serotonin 1s receptor structure came out. It was actually predicted by the biochemical experiments that it might have a bound cholesterol molecule, which has been verified by other groups by the crystal structure, which came out very recently, I think in 2021. So here is the structure, and this is for the serotonin 1 receptor, which has got the bound cholesterol molecule as in the form of a non-annular lipid. All right, so there is another way by which the lipid environments modulate protein environment, or proteins modulate lipid environment. So when we say lipid-protein interaction, it is actually a mutual effect in the sense that lipids influence the function of protein or the dynamic features of the protein. Vice versa, proteins will also influence the dynamics and functioning of the lipid molecules. And that's what is broadly termed as lipid-protein interaction. So one of the form of lipid-protein interaction is hydrophobic mismatch. So what happens in hydrophobic mismatch is that, so typically when we have a transmembrane domain, that means that it has got a hydrophobic amino acid sequence. So they are not very stable when they are exposed to aqueous environment. Now it may so happen that for some reason, due to metabolic dysfunction or some trafficking, the protein is in an environment where the hydrophobic thickness of the surrounding lipid molecule is either less or more than the hydrophobic thickness of the transmembrane protein. And this is broadly termed as hydrophobic mismatch. Now there will be two consequences here. When the hydrophobic thickness of the surrounding bilayer is actually smaller than the transmembrane domain of the membrane protein, then what will happen is, since there are exposed areas in the protein which might not be very stable when it comes in contact with water, so what will happen is this protein will try to tilt itself. So that's one way of avoiding aqueous exposure, or the protein will try to aggregate so that the exposed part are minimized. Both have been found in example. For example, it has been very well studied with gramicidin because the kind of manipulation that I was telling you about during hydrophobic mismatch can be very easily carried out in gramicidin. And since gramicidin is a ion channel, its function can be very easily found out by electrophysiological measurements. So a lot of hydrophobic mismatch data has been obtained using gramicidin as a model system, right? Now, so as I mentioned, that two situations can arise where the hydrophobic thickness is smaller than the transmembrane thickness, or the hydrophobic thickness is actually more than the hydrophobic thickness of the transmembrane protein. So in the latter case, what will happen, that is that from the protein point of view, um, nothing will happen if the hydrophobic thickness is more than the transmembrane domain of the protein. Proteins will be fine. The lipids will have to go some kind of modification. So this is shown here. For example, if the transmembrane domain is actually shorter than the hydrophobic thickness of the lipid molecule, what will happen? The lipids that are actually in the immediate vicinity of the transmembrane domain, maybe those annular lipids, will have to adjust themselves so that they are in the close contact with the transmembrane domain. So in other words, their flexibility will be, or their mobility will be slightly different compared to the uh, compared to the lipids which are distant. So here, for example, the extent of lipid order will decrease because these lipids will be more ordered because they are in the vicinity of this lipid molecule. And as we go further, the sorry, yeah, as we go further, the order will change. Likewise, if we have the other situation, for example, the hydrophobic thickness is actually shorter than the transmembrane domain of the uh, uh, protein. One way to adjust that is that the protein will tilt or aggregate. The another way of adjusting that is that the lipids will undergo some kind of modification. So the lipids that are in the close vicinity will be more ordered. On the other hand, the lipids that are away from the transmembrane protein will be less ordered. So these, uh, situation of hydrophobic mismatch is very relevant in case of sorting. So, for example, the hydrophobic thickness of ER membrane is actually much shorter compared to plasma membrane. So the resident ER proteins will have a shorter transmembrane domain compared to plasma membrane domain. So if there is any uh, you know pathological situation which is sort of interfering with the sorting, so instead of a protein which is supposed to be in the ER, and if that protein is now found in the plasma membrane, it will experience hydrophobic mismatch. So some of the altered physiology will be due to the hydrophobic mismatch of the ER protein in the plasma membrane. Some of the Golgi protein may also, experience similar situation. All right. So I told you that there are many ways by which the membrane physical properties actually influence the functioning of membrane proteins. So I thought of giving one particular example from my own research, and that has to do with the um, again a serotonin 1 receptor, and as I already mentioned earlier that although cholesterol is known for although cholesterol has been shown as a non-annular lipid for serotonin 1 receptor, but there are other features that or other factors that cholesterol modulates, which also have some indirect effect on the functioning of serotonin 1 receptor. And one of that form is a form of membrane potential called dipole potential. So without going into much detail about what dipole potential is and how is it exactly measured, what has been found is that cholesterol has very profound effect on this form of membrane potential compared to its immediate precursors. That is 7-dehydroxycholesterol, desmosterol, coprastenol, and aldosterol, which is a sterol analog that is found in lower eukaryotes like Drosophila, for example. So we were interested in finding out whether this particular physical property has any effect on the functioning of serotonin 1 receptor or not, and if it is so, then how can we modulate that, and again, how can we restore the activity of the receptor if we modulate the uh, dipole potential. So for that, we have taken a natural source of serotonin 1 receptor. This is found in the hippocampal region of the brain. So we got bovine hippocampal tissue from which we derive the membrane, which is a rich source of serotonin 1A receptor. And what we did is we selectively took out cholesterol using methyl beta cyclodextrin, which is a drug which can selectively take out cholesterol, leaving other phospholipids back in the membrane under specific conditions. Similarly, we can also put back some of the sterols or cholesterol at again at specific conditions. So, and we have the ability to measure cholesterol concentration, and the property that I was interested in is the dipole potential. So I also measured the dipole potential under the conditions where the cholesterol concentration was changing. What we found that the dipole potential steadily depends on the cholesterol content in the membrane, and showed a very nice correlation. Interestingly, what we found that the functioning of the lipid serotonin 1A receptor, that is ligand binding in this case, specific ligand that is appreciated 8-OHD pack, and that ligand binding, which is considered as a function of the receptor, was also dependent on the dipole potential. Now, since cholesterol has a profound effect on dipole potential, we thought that if we take out cholesterol, and that means we will be able to change the dipole potential of the membrane. So if dipole potential is a profound influencer of serotonin 1A receptor, we should be able to capture that in the activity assay. So we could show that the activity of serotonin 1A receptor actually depends on the dipole potential over a wide range of, over a narrow range of dipole potential. Now, what we then did is we took out cholesterol using methyl beta cyclodextrin, activity came down. But when we put back cholesterol, we actually got activity back. So that means dipole potential is one of the factors in influencing the activity of the serotonin 1A receptor. But then we also did something else, which is based on the fact that if you take a precursor of 7-dehydroxycholesterol, which I have already shown has a very reduced effect on membrane dipole potential in this graph. So when we put back 7-dehydroxycholesterol using the same technology of methyl beta cyclodextrin, what happened is the activity got back, but not to the same extent as, that dipole potential got back, but not to the same extent as cholesterol did. So concomitantly, that the activity also came back when cholesterol was put back using methyl beta cyclodextrin, but when 7-DHC was introduced in place of cholesterol, the activity didn't get back to the same level as it did for cholesterol. So that means this suggests that when we depleted cholesterol, we reduced dipole potential. We put back cholesterol, we restored the dipole potential, the activity came back. But when we took another precursor of cholesterol, which has lower dipole potential than cholesterol, the activity did not, the activity was not restored. So in these results suggest that, the physical property of this membrane, in this case, the dipole potential, has a profound effect on the activity of a serotonin 1A receptor. Now, this concept can be borrowed for any other molecule which has the ability to modulate dipole potential. For example, lipid anesthetics, which are molecules which has got permanent dipole moments, have the ability to modulate dipole potential. They will also have the ability to alter the activity of serotonin 1A receptor, and presumably it is through the change in membrane physical properties, that is dipole potential. Because anesthetics are general molecules, they don't bind to any particular protein, yet they exert their function. So one of the hypotheses in the field is that they are modulating the physical properties of the membrane, and that in turn modulates the activity of some of the membrane receptor, for example, serotonin 1A receptor. So this is just one example by which I showed that how physical properties of membrane or lipid membranes modulate the activity of membrane proteins like serotonin receptor. There are many other examples that are found and constantly being discovered. Of course, I don't have the time to go through all of them, but I would encourage you to go through the literature and make yourself familiar with that. All right, I think I should stop here and start taking questions. Santoshji?
So I will just go through if there is any questions from the audience. For the sake of audience, like I was just trying to pose a question, like means for the FCS or the FRAP, like say the diffusion coefficient means diffusion mobility, which is correlating to that of your Brownian motion.
Yes.
Correlating to that. So means under what conditions there can be a limiting factor for such tools?
So they are all in the equilibrium condition. So that means see, these experiments are done in live cells. So if you so that means you are ensuring that they are in some sort of a dynamic equilibrium. Because the reason is we want to correlate diffusion coefficient with the signaling. So if you are measuring diffusion coefficients in fixed cells, you may not be able to do that.
Of course,
So all the Brownian motions will be seized.
Exactly.
Proteins and lipids do move to some extent in fixed cells, but not necessarily will have all the dynamic features.
And also we have to define that a window, like say the volume for the confocal for the FCS or for like say the selective bleaching. So means if the membranes are going to be restricted by any means, we may not expect to see the like say the diffusion, the tool like say the outcome, because it's all like say going to be like say the integrative behavior.
Yes.
Whatever we see. So I would also participants, if they have any questions, just feel free to drop in the chat box. Most of the questions I see, I think we have already answered. It was from Dr. Sudhir Sinha. Like, I think probably you already answered them. Any other questions? I don't see any. Students are saturated. I mean, how close it's going to be, but I think, like, say, it's a very wonderful. I see one additional question from Dr. Sinha.
Yeah, so I agree with that. Peripheral proteins may form complexes with integral proteins. In fact, a classic example of G protein coupled receptors, because G proteins are actually peripheral proteins, which gets attached with the receptor. So yes, that partly makes it even more difficult to purify GPCRs. And when you are reconstituting GPCRs, you have to selectively reconstitute the G proteins also, because they're after all the G protein coupled receptors. Yeah, I completely agree with that.
One thing I was just trying to add, because since you have like say very wide experience, so it means if you can discuss if today or if tomorrow, like something about like different model membranes, like say.
Oh yeah, sure.
Yeah, because that's going to be more helpful in and other than the tool, because other than the microscopy or like say the spectroscopy tools, there are different model membranes also like say discern the ability to means how to approach for the such membrane biology studies.
Definitely.
So yeah, means whenever you have like if not 1 or 2 slides, that would be good enough, like say for our participants here.
Yeah, I'll do that. I didn't want to put too many things in one slide.
Exactly. Yeah. I know, like say it's a very, we understand like means a 1 week like say covering cell biology and the membrane biology, which is like say very integrative and like say very wide field.
Definitely.
But we just want to give like say the means what are the basics and then how the field has been evolving in this particular in both the membrane and the cell biology field. So
sure. Because I think you already introduced some of the membrane remodeling things, and
not yet exactly. I have not gone in much detail, so that will be like say definitely the modeling part. I think you will be trying to cover tomorrow.
I will do that.
I will
when
try to cover from the cell biological
okay,
perspective.
so when I'll be talking about fusion, I'll also talk about little bit about modular membranes. Definitely.